Okay, welcome back. Last time we talked about microscopy, and the time before that we talked a little bit about cell theory. And so what we're going to start today, or in this video I guess, is taking a look at different types of cells and differences between types of cells as a lead into talking about structure and function. Now before we get going with this, I want you to take a few minutes, 5 to 6 minutes is probably enough, find a piece of notepaper and create this simple table here. And two columns. On the left, I want you to make a list of the structures or features that a cell must minimally have to be considered a cell. And then on the right, I want you to write down what the function is of the structure or feature that you wrote on the left side. Now you can make this a little bit more complicated by adding additional columns, and some of you will do that as you realize that this is actually a very simplified form. So pause the video and work on that table, and then so what I'd like some of you to do is post your tables in a discussion, and we can get a discussion going around what this table should look like, rather than me just listing these things off. Okay, so let's take a look at some basic cell types. Now, there are two basic cell types that we have to consider in terms of structure and function. First of all, let's deal with the prokaryotes. Now, we're not going to get into the structure and the functions of the structures of prokaryotes in much detail. We're going to leave that to microbiology. We're going to deal more with eukaryotic cell biology. So let's just touch a little bit on the prokaryotes. Now, the prokaryotes are broken down into two main categories: the bacteria and the Archaebacteria. Now, we'll come back and I'll talk about Archaebacteria momentarily for maybe one slide. But we're going to focus mainly on the bacteria. Now, the bacteria is a hugely diverse group of organisms. Some of them cause disease, but the majority of bacteria are completely harmless. And in fact, without them, your body wouldn't function. You have 10 times more bacterial cells in your body than you have cells of you. So that's an amazing number of cells which are not you in your body. And without them, you wouldn't be healthy, you wouldn't function properly. So bacteria are important in our bodies. They play huge roles in the environment, in maintaining carbon balance and maintaining the flux that we see through a number of nutrient cycles. So try and get over the fact that bacteria generally in the press get a bad rap. A couple of bacteria you've got here on the right, those are some Staphylococcus, and on the right, that's the anthrax bacteria, Bacillus anthracis, which causes anthrax. There was a scare a few years ago of that being sent through the mail to some federal buildings. So let's just take a quick look at a typical bacterial cell. And the first thing you've got to take away from this is that bacteria are massively diverse in terms of their physiologies and their structure. And so this is a generic bacteria. If you look at specific examples of bacteria, you're going to see some of these structures, but not all of them. And then if you look at a different bacteria, you're going to see some different ones. And so the structures you see are reflective of the environment in which a particular bacteria lives and which it's adapted to. So these are some of the things you will see in some bacteria, and not all bacteria will have these structures, and some bacteria will have different structures. So this is just a general introduction. Let's start inside. So all cells, including bacteria, have cytoplasms. The cytoplasm is the grey stuff here, and the cytoplasm is contained within a cell membrane, which I'm going to often refer to as the plasma membrane. And so the plasma membrane basically separates the outside world from the inside world of the cell. And we're going to talk about membrane structure and function in a lot more detail in our next module. Now, outside of the membrane, bacteria have cell walls. Now, even that, which is generally accepted that bacteria have cell walls, there's a group of bacteria called the mycoplasma, and they don't have any cell walls. So, you know, that's kind of, again, an exception. And this is the thing about microbiology, which makes microbiology so tough. And I teach microbiology as well. So the problem with microbiology is that if you think that general biology is difficult because it's so diverse and there aren't always these concrete rules, microbiology is even worse in that respect. It makes it fascinating as a subject, but it makes it even worse because you've got all of these diverse ecological niches or niches where bacteria can live. That means they've got diverse structures and physiologies. So generally speaking, we're going to have a cell wall. Now, the structure of that is different than a plant cell wall and different again from a fungal cell wall. But there's generally a bacterial cell wall made of a molecule called peptidoglycan, which contains proteins or pieces of protein and sugars. So outside of the cell wall, you may find a structure called the capsule. And again, a lot of bacteria aren't going to have a capsule, but if you find it, it's kind of a gelatinous structure. It helps the bacteria attach to surfaces it's going to grow on, and also protects it against desiccation and predation by your own macrophages and also certain protozoa, which actually will engulf and consume certain bacteria. Now, all of those fuzzy little projections are pili. And when you get to physiology, you're going to look at structures called villi, and those are very, very different things. So these are pili. These are like fuzzy hair-like projections. They're made from a single protein called pilin, and they cover the surface of the bacteria if they're present. There may be hundreds to thousands of these. And so those are for attachment, and sometimes they twitch a little bit, and they can actually be used for a form of motility as well. Some bacteria are highly motile, and those bacteria will either have one or many flagellum. Now, the flagella is very different than the flagella you're going to be familiar with probably in structures like sperm cells. So the flagellum here is very different, but structurally very different, functionally very similar. A sperm flagella, it does this business, and it's used for swimming, so it's used for motion, whereas the flagellum on a bacteria does a more of a corkscrew motion, but still is used for motility. So there are some of the structures that you're going to see in a bacteria. Now let's just turn our attention really quickly to the Archaebacteria. So the Archaebacteria are structurally and physiologically going to be very, very different than our bacteria. They're going to have some structural similarities and some physiological similarities. But if you look at the evolutionary origins of the Archaebacteria, they're more closely related to your cells than they are to bacterial cells. So go back and look at that phylogenetic tree with Luca that we looked on, and you'll find that the branches for the eukaryotes and the archae are actually closer together than the branches are for the eukarya and the bacteria, or the Archaebacteria and the bacteria. So Archaebacteria are pretty famous for living in places which can be really inhospitable. And so in this respect, they're called extremophiles. So on the left, here's a pond from Yellowstone. And I think this is Rainbow Pond or something like that. Now, the middle of the pond is too hot for even these extreme Archaebacteria. But around the fringes, you get these populations of extreme Archaebacteria, and they can grow in these extreme temperatures. You know, it may be 90 to 100 degrees Celsius around the fringe of the pond, and the pH will also be very, very low, so very acidic. And they produce this whole range of different pigments, and it's not entirely sure what those pigments are for, but you get these very beautiful colors around the edges of some of these ponds in these hot springs. Now, those bacteria have got some remarkable adaptations in terms of how they can survive very high temperatures. If we put one of your cells in a boiling beaker of water, it's going to fall apart, and that's because the membrane is basically going to melt. Now, the membranes, like we talked about briefly before, is made of lipids, and so lipids melt at certain temperatures. So your membranes will basically melt. Those Archaebacteria have structural adaptations to their membranes, which means they can live there, whereas you can't. So please don't think that eukarya are better and the Archaebacteria and bacteria are inferior. In actual fact, bacteria and Archaebacteria can do many, many things that your cells cannot. In that sense, they're ecologically much, much superior because of their flexibility and where they can grow relative to where we can grow. Okay, on the right is what we're looking at there is an acid mine drainage. So in acid mines, because of the way that iron and some other minerals leach out of the soils during the mining process, you get very, very rapid and very extreme drops in pH. And so that water that's flowing through that stream is probably about pH 1. So that's lower than the pH of the stomach acid that's in your stomach. And the orange there is due to the iron that precipitates out of the water at low pHs. And so there are bacteria, Archaebacteria, growing in that water. And again, they'll have very specific adaptations to keep their internal cytoplasmic pH just right, because you've got all those protons at very low pHs, tons of protons outside the cell. They're going to be trying to get into the cell like gangbusters because of the size of the concentration gradient. So that's just a little bit about Archaebacteria, and don't spend too much time on them. They're just a fascinating group of organisms. We're going to spend the rest of our time looking at eukaryotes and eukaryotic cell structure. So eukaryotes come in kind of 4 flavors, if you like. Animals, typified here by penguins, fungus, typified here by this basidiomycete, this kind of toadstool that you're familiar with if you've ever played certain video games. Plants, typified by the grass in the bottom left, and then protozoa, protists, including protozoa in the bottom right, and that's a Volvox colony, and you'll see those in lab, assuming that we don't kill them like we usually do. So, four groups, four kingdoms of eukaryotes. There are some similarities between their cell structure and cell function, but then there are significant differences as well. And so one of the things I want you to focus on are similarities and differences between cell types. So again, I want you to pause the video in a moment and complete another table which will help you compare and contrast different cell types. Simple column, a simple table, and we're just going to have 3 columns, and in those columns on the left, things that you find in eukaryotic cells. Now, try and do this without going and looking up the answers in a book. What can you do from your head that you already know? That's going to make your brain work, and when you get stuck, talk to a friend, put something in discussion, if you have to, look something up in your book. Okay, present in eukaryotic, but you don't find it in prokaryotes. In the middle, the reverse of that, present in prokaryotic, absent in eukaryotic. And then on the right, present in both types of cell. That's going to be the list of things which are common to all cells, so things all cells must have. So stop the video and see how you get on with that. Okay, so same thing as with our first table. Do what you can, and then take a photo of your table and post it in a discussion. And let's get some people talking about what should go in each of these columns and why what some people put in there may be wrong, and how we can adjust this table. Okay, so let's finish up here with a few quick questions. Some of these are not covered in this short video, but some of them you should know the answers to. So here's your first question, and I've got 3 questions. Let's see how you get on with these. Here's the second question coming up now. Here comes your third and last question. Okay, so that wraps up our discussion just on the 2 types of cells that we see, prokaryotic cells and eukaryotic cells. And I will see you next time when we start looking at some structure-function relationships in eukaryotic cells.